Then this chapter is going to be about viruses, viroids, and prions. So, let's see. Click here first. So here we have an image of hepatitis A virus in making a film. So this virus is one of the viruses that can be preventable through vaccination. However, it has caused some outbreaks lately because we have clusters of population who don't vaccine for whatever reason against this virus, and this one causes hepatitis, of course, but it's easily transmittable. So in this chapter, we're going to see again what are the general characteristics of viruses, and we're going to see the mode of replication as well, and we're going to differentiate viruses from bacteria. So viruses, they don't have their own organelles. Mostly they don't have their own enzymes, few of them have, and that forces them to live inside the cells. So that's why they are intracellular parasites. And then viruses, as opposed to bacteria, which has both nucleic acids, they only have one nucleic acid. It can be DNA or RNA, and there is a major subclassification of viruses based on this. So you can call them, well, DNA viruses, RNA viruses. And then we will see within these groups of viruses some of the differences as well in terms of whether they are single-stranded, double-stranded, if they're segmented, if they're enveloped or not. No matter if you have a DNA virus or RNA virus, this nucleic acid is always protected by a protein coat. This will be the capsid. And the capsid is made out of subunits that we call the capsomeres. And this capsid basically is what it gives, for those viruses that don't have an envelope, a nice three-dimensional shape that you can identify really easily in transmission electron microscopy or in scanning electron microscopy. So then they need to live inside a host cell, and they multiply within the cell using its machinery. So they don't have ribosomes for sure, and then they don't have a mechanism to generate ATP, so they don't have mitochondria. So here we have a table comparing viruses with bacteria that is typical and atypical bacteria. So in the classification of bacteria that has rickettsia and chlamydias, we have these intracellular parasites that are similar to viruses, but the rest of the bacteria, they are not intracellular parasites. And then these two have a plasma membrane, while viruses, they don't have a plasma membrane unless they are enveloped. I mean, they're supposed to put here in the table yes and no. And then reproduction, binary fission, both of these. Viruses, they don't reproduce by binary fission. Depending on the size of the bacteria, it is whether it can pass through a bacteriological filter or not. Some of these bacteria, like chlamydia, they're very small. They can pass through these bacteriological filters that has pores like sometimes up to 0.2 microns in diameter. And then viruses, yes, they pass through these bacteriological filters because they are very, very small. And then only bacteria has both DNA and RNA, while viruses, they don't have. And then some bacteria don't have the mechanism to produce ATP in the class of chlamydia and rickettsia, while the rest they do. And then again, viruses, they don't have the ability to produce ATP. Ribosomes, bacteria depend on them to produce their own proteins. Viruses definitely don't have it. And then sensitivity to antibiotics. When we are talking about antibiotics, we refer to antibacterials. So in this case, yes, they are sensible bacteria to antibacterial drugs or antibiotics. And viruses, we have, I mean, if you call these chemicals that kill the viruses antibiotics, well, we have antibiotics that are antiviral, that are specific to viruses. And then we will talk about the different classes of antibiotics a little bit more. I remember that I talked about it a little bit for bacterial cells and also for viruses like the analogs of nucleic acids, but we have a dedicated chapter for seeing some differences. And then sensitivity to interferons. Interferons are naturally produced antiviral chemicals that we produce when we have viral infections, but viruses are only susceptible to interferons, but not bacteria. Now, viruses have a specific host range that it is specific. So we have viruses that infect a certain number of hosts. Sometimes it will be only one. And the reason for this is because this host has receptors to bind these viruses. And the other hosts, they don't have it. So that's the specificity. So another reason why these viruses can infect a particular host, well, is because maybe this host has a factor that these other hosts don't have. And in terms of a major classification, whether the viruses infects bacteria or animal cells, we have this subclassification. So we have some viruses that infects only bacteria. This will be the bacteriophages. And this receptor for these bacteriophages, which we call them for short phages, they can be found either in the bacterial cell wall, in the fimbriae, or the flagella. So it can be an external appendage in the bacteria, or it can be, of course, in the cell wall, the receptor for the phage. And then in terms of animal viruses, we have receptors that are typically found in the plasma membrane. So here is the different sizes for the viruses. They go from nanometers, which is like between 20 up to 100, or, well, yeah, up to 100. So here we have, what is it? No, this is not a virus. This is a prion, sorry guys. So it will be from 24, the range of size from 24 up to 100. The smallest viruses, well, we have these bacteriophages like that range between 24, and then we have another bacteriophage here, which can be 225. So that's the smallest virus, the bacteriophage, like MS2. And then when it comes to an animal virus, the smallest will be poliovirus. Poliovirus infects the nervous system in the host, and it measures 30 nanometers. This is part of a family member of viruses that are called picorna viruses. Pico for small, RNA or RNA for being an RNA virus. So we will see the families later on. And then here we have coronavirus. And then we have here rhabdovirus, which measures 170 nanometers in length by 70 in width. And it looks like a bullet. And then we have here tobacco mosaic virus, that it's a virus that infects tobacco plants. It measures 250 by 15 nanometers in diameter. And then we have viroids, which are incomplete viruses, I would say. They are only short sequences of RNA, and they are linear in this case. And then we have large viruses like vaccinia viruses. They have like an odd shape. They measure 300 by 200 by 100. So they're almost like if you were like a cube. And then we have Ebola virus that looks like a sugar, like candy cane. It measures 100 nanometers if you extend it. Now, if you compare all of these virus sizes, some of them, like in the case of Ebola, they can be larger than chlamydia, which measures 300 nanometers in diameter. And then here we have E. coli, that it is larger than most of these viruses. And then if you compare all of these viruses and bacteria to one red blood cell, which measures in diameter like 8 micrometers from side to side, well, viruses are very tiny. And then we have hosts for viruses that are plant cells, bacterial cells, animal cells, and even fungal cells. So viruses can infect everything, all the hosts. So now we're going to see the differences between envelope and non-envelope viruses. So when it comes to naming viral particles, if your viral particle is complete, it is called a virion. That virion includes the envelope for viruses that has an envelope. And then a virion will have to be complete a nucleic acid. Again, can be DNA or RNA. And this nucleic acid can be single-stranded or double-stranded. And then the DNA or the RNA, no matter if it's single-stranded or double-stranded, can be linear or circular. And in the case of some viruses like influenza, which is an RNA virus, this RNA virus is segmented. It can have up to 7 segments. And then, again, this virion, if it's a non-envelope virus, will have only a capsid, a coat of subunits made out of capsomeres. And then the envelope itself, it is plasma membrane. It is made out of phospholipids with proteins and carbohydrates. And not all the viruses are enveloped. So the viruses that are not enveloped, you typically call them naked viruses. And then no matter if it's enveloped or not enveloped, the viruses, they have spikes that can protrude out of the capsid or out of the envelope. And these spikes are used to actually attach into the host cells. And the envelope of viruses, it is actually acquired when the virus is exiting the cell, the host cell. So basically the envelope, they get it from the host cell. And now, in terms of a structural protection, the non-envelope viruses are more resistant to chemicals like ether or alcohol or detergents. The proteins that makes the capsid makes them more resistant. And then the envelope viruses, because they have these phospholipids can be dissolved really easily with solvents like alcohol or ether or detergents. I always compare these to some people like me. I typically wear an extra piece of clothing because I'm always cold, guys. You take it out and I'm going to be like shivering like that. So I'm like an envelope virus. So you are more susceptible the more layers that you have, in a way. So then when it comes to the number of nucleotides that a virus can have in their nucleic acid, independently of which one will be, it can be up to 250,000 nucleotides. So their genome is pretty much small for the most part. And again, you only have one of these two, DNA or RNA, never both. And then circular, linear, or segmented, double-stranded, or single-stranded. Now, when it comes to the spikes, they're made of carbohydrate and protein. And then we have some names for some of these, like in the case of the glycoproteins that HIV has. We have GP120, we have GP41, and then we use them to identify the virus. So here we have the shape, the morphology of a non-enveloped polyhedral virus. Polyhedral refers to having more than 12 facets, or around 12 facets in, or angles, I guess, in geometrical shape. So these would be the capsomeres of the virus, and then here we have the nucleic acids. So you can see here difference in color in this transmission electromicrograph. The capsomeres and will be here, these little protrusions making the capsid, and the nucleic acid is found inside the capsid. Here we have another envelope virus. So this, in this case, this is helical. So you have this nucleic acid inside, and then the envelope protecting the virus outside of this capsomere. And then you can see the spikes in this TEM. So for general morphology, the helical viruses, they have a hollow portion that allows for the for the nucleic acid, of course, to be protected inside. And then examples of helical viruses will be rabies and Ebola. And then polyhedral, it has, instead of 12 facets, it has 20 triangular facets and 12 corners. And this is called, more specifically, icosahedron. So they have like 20 triangles, and then they have 12 corners. Examples of these icosahedron viruses with their shape will be adenoviruses and poliovirus. Adenoviruses causes several infections, not only in humans but also in mammals like cows. In us, causes very frequently rhinitis, which basically is an inflammation of the nose. So it causes cold. Many cold viruses are adenoviruses. And then poliovirus, it is a virus that infects the central nervous system, the anterior horns of our spinal cord. And then for envelope viruses, mostly are spherical. And this is because since they have an envelope, the envelope, it is flimsy, so you can take this kind of shape. And then some viruses, they have a complex shape, like in the case of bacteriophages. They look to me like they have a head, they have like a little tail, and they have a base plate. You will see when they are infecting bacteria in the next slide, that for me looks like if it was like an alien ship. Of course, I don't believe in aliens, but I watch movies about them. Uh, so here we have an example then of this, another example of these helical viruses, like Ebola. And then if you make a segment of this piece, you magnify it, you can see that you have a hollow core where you keep this nucleic acid. Here is the bacteriophage. Looks like they look like an alien chip. So this is the head. The head measures side to side 65 nanometers. And then inside the head you have this DNA. And then you have this sheet where you have this protein that protects the internal part of the sheet. Then this base plate with these tail fibers. And then the base plate has some pins. So this is a transmission electron micrograph showing you a bacteria with 3 bacteriophages. Something that you can see, it is part of what they do during the infection phase, which is the attachment part. In this case, this virus has its DNA inside the head, and this one partially has DNA because it already started to inject the DNA into the host. So the DNA passes through this part. And then in this case, this bacteriophage in the middle, it completely emptied its DNA. So that's why you see this dark part, because it doesn't have the DNA. The DNA already went into the host. And then we have here another complex virus like orthopox virus. It has a shape like if it was, from my perspective, like if it was a football. So now let's look into what are virus species, and then we're going to see examples of family, genus, and common name for viruses. Taxonomy. So we have this taxonomy that is based on the Baltimore classification system, in which you will have these names that you give to the viruses. The genus name ends in virus, that will be the suffix. And then you will have family names that end in Viroids. And then you have the order name that ends in ales. Now, a virus species will be a group of viruses that shares the same niche, and then they have similar genetic information. And in this case, it's not like what we do with bacteria, that we follow the classification according to the ribosomal RNA signature. In this case, can be, of course, based on DNA or RNA similarity. So again, genus names ends in virus, family names in Viroids, order names in ales. So now we're going to see the way you culture bacteriophages, and then how you culture viral particles that infect animal cells, and then we're going to list 3 techniques to identify viruses. So if your virus that you're studying, it's a bacteriophage, and you know that the viruses that are intracellular parasites, you have to provide bacterial cells. So you would have a host, most likely is going to be a bacteria like E. coli, sometimes can be Salmonella, but the most typical one that we use is E. coli. So then you will grow E. coli in a Petri plate, And then you will have this virus. Either you can apply it directly into the plate once you grow it together with this E. coli, or you can have them in a test tube and then put it in a lawn of of agar. And then you incubate them both for 24 hours, and then you take out the plate from the incubator and you read the results. What you're going to see in your plate is a series of gaps in the growth of the bacteria that we know as plaques. And then you will count them. And then each plaque will correspond to a single virus infecting your bacterial cell, and this will be called PFUs, plaque forming units. So this is the top view of a part of a plate. So this is the plastic edge of the plate. And then each of these gaps is absence of growth of bacteria. So this is bacteriophage destroying the bacteria. And then all of these that you see here, these beige color, is bacteria that hasn't been infected. And then when you grow these, typically these plaques, they give you more or less similar size in terms of the absence of growth, unless you have some that are clustered together, like these ones, they're clustered together. But these tiny ones will be the most typical size. And the edge of the plaque will be round, typically. If you see a really large plaques that has irregular borders, more than likely is contamination by fungi. Now, for growing animal viruses, you can use live animals like mice, rabbits, guinea pigs. And then for embryos, you can use embryonated egg cells. And then when it comes to embryonated egg cells, you can inject the virus into different parts of the egg, and then you can study the effects in both either the embryo or either in the live animals. And then something about these viruses that are grown in these embryonated eggs, it is used to produce some vaccines. And then that's why in some vaccinations, they always give you like this warning, like if you have allergies, for instance, to albumin or certain proteins of the eggs, you cannot take those vaccines because they are deriving from embryonated eggs from chicks. So here, for instance, we have the example of where you can inject these viruses. So just like as when we grow bacteria in the lab, we put bacteria into certain media, you call inoculation. Well, injecting the viruses is also known as inoculation. So you can inoculate the viruses into the yolk cell. You can inject it into the allantoid or into the amnion or into this membrane, the chorioallantoid membrane. And then why would you inject it in different places? Well, because again, you have receptors in certain areas that are not that are specific for the virus that you would be growing. And as well, we can have cell lines. So we have primary cell lines that are basically kept in the lab by either freezing them and then you put them in flask that has this media that specifically can help you to grow these cell lines, and then you incubate them together with the viruses and see if the viruses produce a specific effect. Now, something that you do when you are transferring these cell lines is that you have them in this flask with the media, and then you introduce like a scraper, cell scraper, that you use it to separate from the bottom part of the flask these cells, and then you will use an enzyme, trypsin, to separate these cells, so that you can grab these cells with a pipetter and put it in a new flask, and then you put this media that typically has antibiotics, antibacterial, and antifungal. And then you grow your cell lines. Now, some cell lines might require a little bit of CO2, so in that case, you put them in an incubator that give you like 5% of CO2, and then you produce with this transfer a monolayer. So basically, these cell lines grow in a single line, in a single layer. And then when you have infection of these cell lines, you study what happens with the cells. And some of these cells, they might form clumps. Some of these cells will die. You can see some problems inside the nucleus or the cytoplasm. No matter what happens, this is called cytopathic effect, or CPE. Now, something about the primary cell lines is that they have a finite number of generations that you can pass them. Some of them, they are long-lived. So in this case, you can grow them, but they start losing their strength, and then they don't grow as well. So you have to make sure that whatever cell line you're using, know if it has a finite number of passes or not that you can give. And then in some other cases, you can use diploid cell lines that are derived from human embryos that can be maintained up to 100 generations. For instance, these ones can be used to grow rabies virus for development of vaccines. And then we have secondary cell lines or continuous cell lines that comes from cancerous cells. For instance, well, in this case, since they are cancer cells are immortal. So these cancer cells derive from human cancer, like in this case, HeLa cells. HeLa cells were donated, I think, like in the 60s by a person who had cancer, Henrietta Lacks. She donated the cancer cells, and then something that you can expect in these cancer cells is that their genome, it is the same, because that's what happened with certain cancers. They just multiply, and they don't change their genome. So an advantage then that these ones can be maintained indefinitely. So here we have an example of how you transfer some of these viral cells, these cell lines. So let's say that you had before a flask, and then you treat it, you scrape part of the cells, you extract them and put them here with some trypsin in this test tube, and then you shake it well to separate the cells, and then you can put them in this new flask with culture medium, and then you would have either monolayer cells in these primary cell lines, or you can have, in this case for these secondary cell lines or cancer line cells, you can have multiple layers. So these are immortal. And they can come also from like lymphocytes. Some cells are derived from lymphomas, and then something about these cells is that they lose cell contact inhibition. Typically, our cells, when they're growing, the normal cells, they touch each other and they stop growing. That's why we don't end up with tumors. But in tumor cells, basically, they multiply, they contact each other, and they don't care. They keep multiplying. So that's a big difference. And that's why you end up with clusters of cells. So here we have some examples of CPE. So these cells, after they were infected by a virus, they form this cluster. This is called syncytium. They form a gigantic cell. So this is a monolayer of cells, for instance. For each of these, you can see the nucleus. So you can identify viruses based on the specific CPEs, cytopathic effects that they create. Also, you can identify them based on the antigens that they can display on the surface, and you can detect those by ELISA. So you have some antibodies that can bind to these antigens, and then when you put some substrate that they can be broken down by this enzyme, it gives you a color change, so that's ELISA. And then the kind of faster way, I guess, and more precise way to identify this will be by using PCR. Depending on your virus, you can use forward or reverse RT-PCR. So now we're going to see how viruses multiply. We're going to see two ways, the lytic cycle and the lysogenic cycle in bacteriophages. And then we're going to see the mode of replication of RNA and DNA viruses. And then we have two types of viruses, the tequatrovirus, virus, sorry, and lambda virus. You're going to see those. It's typically called T virus. It's the even viruses. Now, viruses, they have to land into a receptor in the cells, and then they invade the host cells. So they, once inside the host cell, they take over the metabolic machinery, and then you can study how many viral particles you form out of what is called one-step growth curve. So here we have a general way to know how many number of viruses you can produce. So if you have some cells and then you infect the cells in time 0, this is the graph showing you time in days and then the number of virions. So let's say that you have added 1,000 viral particles. Within 2 to 3 days, these 1,000 viruses, if you're studying the liquid or the media where you grow these viruses in the lab, you will see within 2 to 3 days that you won't have any viral particles. And then after that 2 to 3 days, you will see that your number of virions increase so much. So then if you study the liquid, the media where you're growing it, you take a sample and you count the number of virion particles, you end up, let's say, with 10 million. In day 7. And then day 14, you have 0 again. but you didn't do anything else to these cell lines that you were growing. So what happened between day 0 to 2? Well, something that happened is that the virus goes into the cells, and then they won't be found in this media, this liquid that you have on top of your cells. So they hide inside where they are infecting the cells. So that's why you end up with 0 numbers. But the viruses, if you take the cells and you grind the cells and then you extract the viruses, you will find them there. So your 1,000 viruses that you use to infect the cells, they are inside the cells. So that period where you don't see anything in the liquid media is called the eclipse period. And then you will have a sudden growth in the number of viral particles, because as the viral particles are being made inside the cells, they will burst and then they will be released into the surrounding media. And this is called the period of acute infection. And then suddenly with time, you will have a decline, just like if it was the death phase of the growth curve in bacterial growth. So similarly here, you have a death phase, and then if you don't do anything, like if you don't add more cells and more media, you will end up with 0. So your virus will die because it won't have more cells to infect. Now, when it comes to bacteriophages, they grow in 2 ways: lytic cycle or lysogenic cycle. In the lytic cycle, you will have the bursting of the cells. That's why it's called lytic, because you will lyse the cells as soon as the cells are multiplying so many of the viruses. So the phage will cause lysis and cell death in the host. Example of a virus that do so is T. even, bacteriophage. Take quattro virus. And then some viruses can adapt as the host cells are dying or they don't have as much nutrients available. So this is called the lysogenic. In the lysogenic cycle, the bacteria will form, the bacterial DNA will be infected, will be hybridized by the phage DNA. So these will be forming what is called provirus. And this can happen by lacking of nutrients by the host cells. So sometimes that can lead to some outcomes like we call phage conversion or a specialized transduction. So here are the stages of viral infection. First one, you have to have attachment of the virus into the host cell. In this case will be a bacteriophage that attaches with the tail fibers into the cell wall. So that's the first step, attachment. Second will be penetration. So in the case of bacteriophage, they shorten their sheath and they insert the pin into the cell wall, and they release an enzyme, lysozyme, to make that penetration to happen easily, and then the DNA will be injected. So it will be similar to like when someone is injecting insulin, which I guess is going to be hard to pass because we have these new devices that they do it automatically, transcutaneously. So then after the viruses have penetrated the host cell, in this case the cell wall, you will start forming new DNA and proteins. So this is called biosynthesis. And then in order to do biosynthesis, then what the bacteria do, it is help their own production of DNA and proteins, because the virus take over the machinery. So that's why it's a parasite. And then once you have DNA and proteins, you start assembling, like if it was a car factory. You have different pieces, you bring them all together. This is called maturation. And then once you have your complete virion, you will release. So you will lyse the cells. And in order to do so, you also use the enzyme lysozyme. Now, something that happens during viral replication in comparison to replication of bacteria, it is that replication of bacteria typically happens, of course, by binary fission, but it happens every 20 minutes. So every 20 minutes, you double the number of bacteria. But when it comes to viruses, more or less, the average generation time is 4 hours. But although it's slower than the bacterial replication cycle, every 4 hours you release millions of viruses instead of doubling the number. So most of the times, it is hopeful that someone who has a viral infection receive antiviral drugs every 4 hours. Not all the time happens, because not everyone knows that happened, but it's supposed to be the ideal time for most of these viral infections. So here we have the stages of the lytic cycle. So here we have your host, E. coli, that it is attached by this T-even bacteriophage. So if you magnify here, you can see how the tail fibers adheres this T-even virus into the host. That's attachment. And then in penetration, this sheet is contracted. The tail, the pin, which is in the middle of the base plate, now it penetrates the cell wall together with the aid of the lysozyme. And then, just like if it was a hypodermic needle, this virus injects the DNA. So it's going into the cytoplasm of the host. And then the DNA will carry instructions for the host cell to start making more DNA during biosynthesis, making heads, making tails, sheath, base plate, pins, tail fibers, and then during maturation, you put them all together. So you put the DNA inside the head, the capsid, and then you put these sheet underneath, and then the base plate, the tail fibers, and these will be ready to go in release stage. Sometimes what you can expect, it is misassembly. Sometimes you can have some capsids that won't have this DNA. And depending on the type of virus, you can have sometimes carrying over part of the DNA of the host together with the viral DNA. And that's how you have phage conversion. So that's for T-even, the lytic cycle. Now for lambda virus, which can undergo the lysogenic cycle, but it can also undergo the lytic cycle. So lambda virus can initiate infection with lytic cycle, and then transition, yes, that's a good word, transition into the lysogenic cycle. So lysogeny gives an advantage for viruses that are non-lysogenic. It helps them to survive more. So I cannot make that. I can do it after. So we have this DNA that it is incorporated into the host cell DNA, and then you form the prophage. And then when the host cell is running out of nutrients, you will have a reduction of this replication, essentially. Because what is the advantage of the virus to kill the host cell if the host cell is going to die and I will die? So I rather wait a period of time just to see if my host cell can grab nutrients, and then I can grow back again happily. And then that's when I can make numbers, and then I can lyse the cell. So here is the combined lytic and lysogenic cycle for lambda. Just as before, for T even, lambda attaches into the host cell, and then in this case you have penetration as well. So they put here the DNA for lambda with this fuchsia color, and then purple for the host cell. So then when this virus starts forming more copies, it can just make their own DNA, form more viral particles in maturation, and then it releases them. That's the lytic cycle, and then infects more cells. But then nutrients are running low. So now this lambda DNA is going to hybridize with this bacterial DNA, and then you form a prophage. So you can see a little piece here of this virus DNA with the host cell DNA. And then when these undergo binary fission, then you make not only the copy of the bacterial DNA, but also of the viral DNA. And then you undergo several cell divisions, and then when the conditions become favorable, then this DNA can excise from the virus from the host cell DNA, and then you can undergo the lytic cycle again. So kind of that's a clean excision, basically separation. No pieces of the host DNA. However, sometimes that excision can lead to bringing part of this DNA into the viral DNA, and then you will have carrying some genes of the bacteria that can be useful to other bacteria to resist antibiotics or disinfectants, or become more virulent in general, no matter which way it can be. So then out of lysogeny, I was kind of getting ahead of myself. So out of lysogeny, which is that cycle where you replicate the DNA of the virus together with the host cell DNA, you can have three potential outcomes. Well, the first one would be that the lysogenic host cells are immune to reinfection from the same phage. So your bacterial cells develop immunity. And if you remember, one way to develop immunity is by having these restriction enzymes. And then the second way, it is by basically the second outcome will be having phage conversion. So then the host cell will exhibit new properties that were encoded by this prophage DNA. So an example would be the production of diphtheria toxin by Corynebacterium diphtheriae. And then the other one would be a specialized transduction, in which a specific DNA from the bacteria will be transferred by the same virus into another bacteria. And then the recipient become similar to the donor bacteria. So here we have some of these examples from this specialized transduction. So here we have bacteria that undergo lysogeny, or a virus that undergo lysogeny. So here you have your prophage, and then you have this bacteria that originally has this galactose gene. So this bacteria can use galactose. And then when this phage DNA excises or cuts out from this DNA from the host, it will now carry the galactose gene, and during assembly, it put it into the head, and then it goes outside of the cell during lytic cycle, infects a new cell. So this was the donor cell, this will be the recipient cell, and then when this phage injects this phage cell, now it will inject this galactose gene, and this cell, the recipient during the lysogenic cycle, will acquire this galactose gene. So this was, let's say, galactose plus, and this was galactose minus. Now it will become galactose plus, and now it can use lactose, or galactose, sorry. So that's a specialized transduction. Any questions? Now, mode of replication for animal cells is the same steps. So you will have attachment, and then entry, which will be penetration, but and you have maturation and release. But in this case, for animal cell viruses, you have to have uncoating. Because when this virus attaches and penetrates or enters into the cell, you will enter into the cell with the capsid, and then you have to uncoat so that you can have the DNA or the RNA ready to undergo biosynthesis. So you can create more copies of DNA or RNA, plus you make, of course, more proteins. And then an outcome of this infection from animal cells is either you can go chronic infection or just acute infection. So here is then a difference between, or differences between the bacterial cell infection and then animal cell infection by viruses. So in attachment, bacteriophages uses tail fibers to attach into the cell wall. In the case of animal viruses, they use receptors in the plasma membrane of the host cell, and these receptors can be made out of glycoproteins, for instance. And then viral DNA, it is injected during penetration into the host cell, while in animal cells, capsid enters together with the DNA or RNA by receptor-mediated endocytosis or fusion. In the case of envelope viruses, they fuse the envelope, and then they penetrate the capsid and the nucleic acid. In the case of the uncoating is not needed in the bacteriophages because directly the DNA it enters into the host cell. And in the case of animal viruses, you require enzymatic removal of the capsid proteins so that you can have the DNA or the RNA in the host cell. Now, since bacteria don't have a nucleus, the replication or the biosynthesis happens only in the cytoplasm. But in animal viruses, and with, well, I'm going to ask you that question, but with few exceptions, but DNA viruses multiply in the nucleus, while RNA viruses multiply in the cytoplasm. And then viruses, of course, again, because bacteria don't have a nucleus, they always multiply in the cytoplasm. Now, before we move on into this other part, so why DNA viruses has to replicate in the nucleus in animal cells? Why they cannot multiply or undergo biosynthesis? Well, why they cannot replicate in the cytoplasm, DNA viruses?
Because that's where the enzymes are that replicate.
What enzyme?
Like the
It's DNA virus, so what do you need?
DNA is in the nucleus, it's not in the cytoplasm.
It will be a DNA polymerase, right?
Yeah.
Because we have the DNA polymerase in the nucleus, that's why. Believe it or not, many people don't know that. So it's kind of something that, unless you kind of think about it that way. I mean, years ago I didn't know that, but then when I started to think about it, and I said, well, why only here? Well, it's the DNA polymerase. So, and you can impress someone with that. So this one can the bacteriophages can integrate into the DNA of the host cell, forming lysogeny cycle or chronic infection. In the case of animal cells, that lysogeny, it is called latency. We can also have integration of nucleic acid into the DNA and form a provirus, not a prophage, a provirus. And then part of this chronic infection can be slow viral infection, and some of the viruses can inactivate tumor suppressor genes causing cancer. And then exit or release, it is through lysis in bacteriophages. And then in the case of animal cells, if it's an envelope virus, it buds out. And in the case of naked viruses, it ruptures the plasma membrane. So there is nothing different when it comes to most of these stages for multiplication of animal viruses, except that you have receptors specific for the viruses that are specific for certain cells, like for instance, hepatitis viruses, they infect only the liver, okay? So because we have receptors for those viruses. So we have a specificity within host. And then release is by budding, and then maturation, it's the same. So here we have the entry of viruses into the host cells. So this is called receptor-mediated endocytosis. So this virus connects or attaches into these receptors, and that triggers endocytosis. And then that will be for this virus that it is a retrovirus. And then herpes virus, it's a DNA virus that has an envelope. So that envelope is left behind when it fuses into this plasma membrane. So this is the plasma membrane of the host cell, and then it leaves it in here, and then it penetrates the DNA. So this is a diagram to show you the budding process when you release a viral particle. So you have this viral capsid. Inside, of course, you have the nucleic acid. This is the host plasma membrane. It has viral proteins. And then, little by little, you start forming a little protrusion that ultimately is going to form the envelope. So this is just plasma membrane wrapping around this capsid. And this happens in this lentivirus, for instance. So for biosynthesis, again, DNA viruses, they replicate the DNA in the nucleus, and then they synthesize the capsid proteins in the cytoplasm. And then kind of something that happens with this DNA virus. So once you make your capsid proteins in the cytoplasm, you assemble this virus in the nucleus. So you bring the capsid into the nucleus. So here we have this example of this DNA virus. In this case, this is HPV. So this virus has this capsid, this DNA, attaches into this host cell, and then it will penetrate or enter in, and then uncode. So then all of these capsid proteins are released, so you have this viral DNA. The viral DNA will be transcribed using messenger RNA to encode early viral proteins, which is basically proteins that can be used to make some of these viral proteins. And then the DNA migrates into this nucleus where you make this messenger RNA, and then you will exit this messenger RNA while you make more copies of the DNA inside the nucleus, and then you make these structured proteins that are going to be forming the capsid, and then all of these copies of DNA that you have inside the nucleus has to be assembled. So you bring these capsid proteins into the nucleus, and then you form this virion, and then this virion will be released. So that's the life cycle of this DNA virus. So for DNA viruses that are double-stranded, so we have adenoviridae that is not enveloped, so it's a naked double-stranded DNA virus. Mostly causes infections in humans, and then we have a type of virus that is called masked adenovirus that infects the udders of cows, and it can form tumors in cows. So this is masked adenovirus, for instance. This is an scanning electron micrograph that shows you the shape of this virus. So it's icosahedral. You can see these capsomeres. And this virus, to be protected by this processing of taking this picture, it was coated with salts, metal salts. And that basically, as this virus is bombarded with the electrons, it won't be destroyed as much because this part is all flattened. So in this case, the electrons basically destroyed this virus. But again, look at the shapes. They're so beautiful. I mean, of course, the effects are not good, but I mean, it's one of the things about microbiology. Yeah. So viruses, they're very neat. Another DNA virus, one of the gigantic DNA viruses, we have poxviridae. That's a family. So they are double-stranded envelope viruses and causes skin infections. Like we have smallpox virus and monkeypox virus, or mpox. So their assembly, in this case for these DNA viruses, occurs in the cytoplasm. They produce a very typical cytopathic effect where they create clumps inside the cytoplasm and also inside the nucleus. So for these two viruses, I mean, this one right now, monkeypox virus, it is a virus that is an emerging infectious disease. It has been creating some outbreaks. Smallpox, it is this virus that thankfully was eradicated around early 70s. And this virus used to cause a lot of infections in the brain, not only in the skin. In the skin, it left a lot of scars in people. And I mean, who cares? You can live with scars, but when it infected the brain, it created encephalitis. Many people die. And again, thankfully, we eradicated with vaccination. And then monkeypox, it doesn't create those severe infections as smallpox, but still is one of these type of viruses that if it can infect a population that is immunocompromised, then it can have lethality.